Can we study human brain development and disease without using the brain of living organisms? I am studying the human brain with an in vitro model called organoids. Brain organoids are three-dimensional aggregates of cells that recapitulate important aspects of human brain development. In my project, I specifically focused on a brain structure called the corpus callosum, which makes the communication between the two brain hemispheres. When the corpus callosum is absent, patients have a disease called corpus callosum agenesis. So, using organoids derived from pluripotent stem cells of patients, I was able to build an in vitro model of the corpus callosum and study disease mechanisms in great detail. My research is composed of three cyclical parts: experimentation, result analysis, and discussion. The experimentation part takes place in the laboratory, and working in a lab means that every day there is a new challenge. A lot of time is spent planning and executing experiments, which in my case means cultivating and differentiating patient stem cells into organoids. This is followed by the analysis and interpretation of experimental results. We use a lot of different techniques like microscopy and sequencing in order to understand disease mechanisms. By the end of a milestone, our findings are discussed with collaborators and colleagues, and new ideas emerge on how to continue with the research. This process repeats itself until the work is ready to be presented to the scientific community. Our work happens at the interface between fundamental and medical research. Organoids are minimalistic models capable of reproducing aspects of human development that are otherwise not accessible, and they are transforming the way we study the human brain. On one hand, the use of patient-derived stem cells makes our research more personalized and is pushing the boundaries for understanding, diagnosing, and eventually treating neuropsychiatric disorders. On the other hand, because these organ-like tissues are generated in vitro, we are effectively reducing the extent of animal experimentation required for neuroscience research. I am fascinated by the unknown, and my research is a way to turn that curiosity into answers. The Vienna BioCenter fosters an environment of scientific creativity and interdisciplinary studies. I was able to build collaborations with medical professionals from the Vienna General Hospital and with bioengineering experts from the University of Tokyo, which were instrumental to achieve our scientific goals. During these years, I was embedded in a worldwide network of scientists while working at an amazing research institute with brilliant colleagues.